Now in this section we will study the scientific method. The process of studying biology is basically based on a scientific study of life, and a scientific method is it's mainly a process that goes on in our minds, and many of us actually do apply scientific methods often in our daily lives. You may not just be aware of the mindset in a scientific setting. So let's just look at what is a scientific method. Scientific method begins with the first set, that is to observe and to collect data. Observations are always made, which is always looking at either a data collection or a certain phenomena that is being observed, or a certain appearance that has been studied. So that's just the observation. Sometimes we use instruments to study them, and sometimes it just could be using your five senses. For example, you've observed a change of color, or you've observed a height of an organism growing into a certain length, and so forth. So it is just the first step in scientific method. Now, after observation, the next statement is called its hypotheses, which is an educated guess. An educated guess means that looking at the observation that was just collected, the scientist will use their sense to somehow relate to that data, and they will come up with a statement, but this statement is not really tested yet. So it's just an input from various sources to suggest a possible explanation to the observation that have been observed. The hypothesis then follows the next step, which is experimenting these hypotheses. And experimentation will always be done after you have done your observation. Now, sometimes observations and experiments will be successful, and if that's the case, then you can conclude and come up with your hypotheses as to be successful, and your experiments could support that hypothesis. Or sometimes your experiments will not support an hypothesis and it will be rejected. And if it's rejected, you'd go back you will actually go back and start your experimentation again. My favorite example for this one is in everyday life is going to a physician's office. When you go to a physician's office, the first thing that the nurse will do is observe and collect data about your blood pressure, your height, your weight, and all kinds of questions. The physician comes in, the doctor is going to use some more instruments to possibly get some more observations. He may be checking your breathing, or or do different test on you while sitting in his office. And after that, he will come to a possible guess or an explanation to what he has observed. And the way he does test that hypothesis is to give you a medication. Now, if the medication works and you feel well, and it's a success story because the experiment was successful. Often, once in a while, you'll get a call from the nurse's office saying that the doctor wants you to change the medication. And that's because they have done some culture study and found that the medication that they gave did not cover that range of organisms, and that is they have just rejected their hypotheses and tested you with a different experiment. So this is the process of scientific method. In this example, this is an experiment in which a reasoning was carried out, in which we will we're pretty much in this example looking at the growth of pigeon pea winter, which pigeon pea which grows very well in India and Pakistan and those part of the world which are more subtropical. And in order to study them at whether they provide enough nutrients to or with enough nitrogen to the growth of the plant. This study is done over a period of 3 years, and there were 4 different groups chosen. A control group is always needed because you cannot do an experiment unless you can have a control group in which you will find, is there an effect of fertilization treatment or not. So as simple as that. So this would be your control group. Your test over here, you had the test spot in which this particular experiment was done, in which they were testing whether the pigeon pea and the winter wheat rotation helped each other to provide more nitrogen to the soil. And then in between we also had 2 test spots, one which had 90 kilograms of nitrogen given, and the other that was given 40 kilograms of nitrogen or the fertilizer. And so you've got an experimentation group and you have a control group. So you can never design an experiment without a control group, because if you don't have something to compare it with, it's very difficult to detect the effect of the feature that you're looking at. So you always, once an experiment is carried on, you always definitely need some sort of a graph or some sort of a data to analyze that observation. And after analysis, either a hypothesis can be proved, or it can be, if the prediction happens, then the hypothesis is unchallenged. If it not, hypothesis is unsupportable. As simple as that. So back to our scientific method. After you've done an experiment in which the one that I just showed you about, the experimental involving the control pots and the test pots of the 3 different types, the results are important, because the results will either you will collect data from the results, and then you will strengthen that the probabilities that came after your results. If the probability is low, then you usually have good results. A higher the probability will make it difficult to dismiss a random chance as the sole cause of the results. So often you will look for standard deviation at the end of a scientific statistical analysis. Again, more terminology into scientific methods. The results are always analyzed and interpreted. So there's no use of collecting data and doing experiments if you cannot sit down and come up with a designed process by which you can analyze that data. And conclusions are what will cause the scientist to think about how they can interpret that results. Now, a lot of times when we do experiments, and you will have this experience in laboratory settings, most of the time students find that if they have done an experiment, it should be successful. But that doesn't really happen in real life scenarios. Most often experiments result in failure. We just don't hear about those failed experiments. But it's important, if you have a failed experiment, as a scientific thinker, you should be able to see what went wrong. That is the most important thing. Once you know what went wrong, you can go back and revise your experiment. And eventually, over a period of time, you will have a success story. And the success stories are the ones that go into scientific journals, and they go for peer reviews, and eventually are all over the internet or the web, and they make other scientists attempt to duplicate them or for their own published findings. But those are all the success stories. A lot of times you don't hear about the things that did not went well during this process. So what is a scientific theory? Scientific theory is mainly taking related hypotheses and making some prediction out of that. Often the scientific theory will be supported by broad range of observations and data. For example, a scientific principle will eventually become a law, and a law is widely accepted theory. The best example that I can give you right now for that is, take any piece of object and throw it with your hand up. The law is that it will fall down due to Newton's law of gravity. That's how widely accepted a law is considered to be. It's something that will always take place regardless of who carries that experiment, and it will be done throughout the same in the same planet. Now here we can see a comparison between a bird and a bat. Now a wing on the bat and the bird on the bird, and wing on the bat and the wing on the bird, they do the same function, which is they help it to fly. But you can see the arrangements are very different. Let's look at the whale, the cat, and the horse, and the human. All these are showing you something in common, and that is that they're all limbs of different sorts. And what will the limb do? What is the principle behind that? And this is all discussed in the chapter when we will discuss evolution. We will look at these different analogies and homologies that are present between the two groups. So what we call as a bird's wings is analogous to the bat, and these are all homologous or homologs traits, because they all have the same feature that they're used for. They're all used for movement. So looking at certain observations, scientists will come up with certain supported statement, and we will go into these two terms later in evolution when we get that chapter. So almost at the end, what types of experimental variables are there? Experimental variables are either dependent or independent. The independent variable is always applied one way to the experimental group, and it's applied a different way to a control group. For example, you can see the independent variable is shown on the x-axis. The dependent variable, that is the variable that is measured to generate data, is usually given on the y-axis. So this is important when you're doing graphing. This is your x-axis. The x-axis will always have the independent variable. So you can see the wheat biomass was the variable that was used to measure that data, and it is always plotted on the y-axis. So a little bit we talked about a lot of different things in this chapter. This concludes this week's lecture.